Okay. So here we go. Let me display it up here. All right, so you guys have to remind me kind of where we, oops, I guess it would help if I unmuted that so you can actually see. So we've already talked about this. No,
we were at membrane proteins and then
alrighty. So we've talked about how phospholipids can move, and then we talked about how the composition of the phospholipids can be saturated or unsaturated. Can somebody tell me just real quickly? I know we're past this, but I want to just review with this. If you have an organism that lives outside, so let's say we're talking about a plant, and we're talking about the cell membrane of the plant, and it's fall and it's starting to get cold outside. How does the composition of the phospholipids, being saturated or unsaturated, play a role in helping the plant to survive? How does whether or not the phospholipids in the membrane, if they're saturated or unsaturated, how does that play a role in the survival of the plant as the temperature drops down? Sage?
Would it change like how it freezes if it would during the winter? Like the unsaturated might freeze at lower temperature than the ones that are saturated?
Yes, absolutely, absolutely. And so therefore, because the membrane that has the unsaturated fatty acids can remain fluid at lower temperatures, it helps with their survival, because the membrane's job is to allow things to go in and out of the cell, and so you have to have exchange with the environment. And if that membrane freezes and becomes solid, you don't have that happening, the things going into and out of the cell, which can cause the cell to die. Absolutely. And so then we talked about cholesterol. Cholesterol, I think this is where we left off, right? Cholesterol? All right, cholesterol was a type of steroid, and I left this up from last hour. Lipids, remember, there's three different kinds: triglycerides, phospholipids, and steroids. So cholesterol is an example of this steroid. And it had a unique shape in that it's 4 fused rings. So when you have this cholesterol embedded in the membrane, again, when the temperature falls and gets cooler, it doesn't allow the phospholipids to pack very tightly together, which also helps to maintain its fluidity. So if it has unsaturated fatty acids as well as cholesterol, helps to maintain the fluid nature of the membrane. All right, that's where we left off on Friday. So in addition to phospholipids, membranes aren't just phospholipids, they also are composed of proteins. And the proteins can also move throughout the membrane just like lipids can. Proteins are much larger than lipids and move more slowly. To investigate whether membrane proteins move, researchers fused a mouse cell and a human cell. So that's what we see here. So this is a very kind of simplistic look at a mouse cell and a human cell. These balls that are different colors are supposed to represent the proteins on the surface of the cell membrane. So when they fuse a mouse cell and a human cell, what they were trying to see is, do the proteins move or do they stay put? And so when they fused them together, if they stayed put, you'd have half the cell have human proteins, half the cell have mouse proteins. And what they found is after an hour, they were kind of interspersed and intermixed, showing that proteins also move through the lipid bilayer. So remember, the lipids, or the lipid bilayer, lipids can switch spots and move through the lipid bilayer, but also proteins can too. So the membrane is really not a static entity, it's constantly changing and constantly moving. All right, so what are the jobs of these proteins within the membrane? The membrane is a collage of different proteins embedded in the fluid matrix of the lipid bilayer, so kind of in the center here. Proteins determine most of the membrane's specific functions. Peripheral proteins are not embedded, and integral proteins are. They penetrate the hydrophobic core and often span the membrane. So when it says proteins determine most of the cell membrane specific functions, if you have two cells can differ based upon the proteins that are embedded in the membrane. So if you have protein A and protein B in this membrane, and protein C and D, protein C and D have different functions than each other, but also from A and B. So these two cells would have different functions and would be able to do different things based upon the composition of the proteins in there. So that's one way that cell types differ from one another. This guy right here that I just drew up here, would this be peripheral or an integral protein? This is integral, all right? So it goes through the hydrophobic core. So remember that we talked about this is hydrophobic, it's phospholipid tails, they're nonpolar. And Friday we talked about how the protein is also polar at the ends, where it touches the water-based solutions inside and outside the cell, and it has a nonpolar interior or hydrophobic interior, just like the phospholipids. And so that's an integral protein. And so that's what this picture is showing you, is these are all the integral proteins. So this part and this part would be the hydrophilic, and this part in the interior would be hydrophobic. This is showing you here a peripheral protein. Peripheral is called that because it comes from the word periphery. The periphery of something, what does that mean? The outside part, all right, the outer limits, all right, of it. And so so it's on the, if we're talking about the membrane, all right, it is on the outer skirts of the membrane. It's not within the membrane, all right, of the bilayer. All right, and so then this is pulling in what we talked about on Friday. This is the extracellular matrix that connects cells, one cell to another, and so on. All right, so let's look at integral proteins. Integral proteins that span the membrane are called transmembrane proteins. This could be integral or transmembrane proteins. The hydrophobic regions of these integral proteins consist of one or more stretches of nonpolar amino acids, often coiled into alpha helixes. So what does this mean? Interior portion is nonpolar. All right, so the amino acids in the interior portion are going to have nonpolar R groups. That's what it means by nonpolar amino acids. So what is this picture showing you here? What is this picture showing? Do you have an idea of what that picture is showing? Roman? Yeah, it's showing you the tertiary structure of a protein. So this is pulling in from chapter 5 when we learned about proteins. So when we draw a protein like this and say proteins are embedded in the membrane, remember what proteins are. They're chains of amino acids. They have the alpha helix and the beta pleated sheet, and then they fold on each other, and interactions of the R groups cause it to have a certain shape. All right, that's what we see here. So this is one long chain of amino acids. So this is the tertiary structure of the protein. It's not quaternary. We would need to have another amino acid chain. You have to have multiple amino acid chains. This is just one. So this is the tertiary structure of the shape of this particular molecule. And so this ribbon is your chain of amino acids. So it starts here, and then it coils all around, and so on. And here's the other end. So that's your chain. Why do they say C terminus and N terminus? Because when you have an amino acid chain, let's just do a quick little review here. An amino acid has a central carbon atom that can make 4 bonds. When we call it an amino acid, we call it amino because it has what group? The amino group, which is NH2. We call it an acid because it has a carboxyl group. It makes it acidic. Every amino acid also has a hydrogen and an R group. And so this is the construction of one amino acid. So this is a chain of amino acids. So if we hook this together with another amino acid real quick, just draw another amino acid here real quick. When we hook them together, what comes off? H2O, right? So this comes off as a molecule of H2O, and this carbon will be bonded to this nitrogen, and we won't have these two anymore. So when you have a chain of amino acids, notice that at one end, you have the amino group, which contains nitrogen. That's the N terminus, the N end. And then you have one end that has the carboxyl group that contains carbon, and that's the other end. So that's why they call it that. All right, and so that's just a chain of amino acids. Okay? Alrighty. And so every protein embedded in the membrane has a different shape, and the shape determines its function, what it's going to do in the membrane. All right, so what kinds of things can a protein do in the membrane? We have 6 different things here. We are going to talk, I'm going to introduce all of these here. Well, the majority of this chapter is going to be we're going to talk about transport, and we'll talk a little bit about cell-to-cell recognition. So I'm going to explain these functions based upon these pictures here. So function A here is transport. We've already talked about how some proteins have a channel in it that allows things to move through and they therefore go into and out of the cell. There's also another type of transport protein that actually changes shape. And so that has a specific shape, and a molecule inside the cell can bind to that, and then the protein actually changes shape, and so that opens up on this end and allows those molecules out. So transport across the membrane is function A. Function B is an enzyme. What do enzymes do? They catalyze or speed up chemical reactions, absolutely. And so here we have 2 different enzymes. How do I know that there are 2 different enzymes? They have 2 different shapes. All right, and so this enzyme is catalyzing one reaction. Real simple, simplistic kind of idea. Red triangle gets converted to blue square. Chemical A gets converted to chemical B. This enzyme speeds that up. Then the blue square, chemical B, gets converted to chemical C by the enzyme, the second enzyme. So those are enzymes. Another type of function in the cell of a protein is a receptor protein. And it says signal transduction. This is a way for cells to communicate with one another. So this signal molecule, I'm just going to, on here, just from the signal molecule here, this molecule comes from another cell. And this is involved in cell communication. So what happens is if you have a cell A here at one part in one part of your body, cell A here makes the signal molecules. The signal molecules go out and usually go into your bloodstream. These signal molecules go into your bloodstream where they can go to another part of your body. This other part of your body, let's say you have cell A and a couple of cells here. This molecule that released the signal molecule is going to communicate with another part of your body. How does it know to be able to communicate with the other part of your body? It has a particular receptor protein that will allow, it has a shape complementary to the signal protein. So here's your signal protein, can bind to this receptor protein, and what that does is cause some kind of change to occur in that cell. And that change, whatever it might be a certain chemical reaction that causes something to be produced or something like that. That would never happen had the signal molecule not reached that cell. And so other cells might have different receptor proteins. Would that signal molecule affect that cell? No. If this cell does not have the right receptor proteins, then it can't communicate with it. So that's what that means, the signal transduction. Yes?
Is this how neurotransmitters work?
This is how neurotransmitters work. So neurotransmitters are a type of signal molecule. Absolutely. All right? And so that's, and hormones work this way. So hormones, a lot of our hormones are produced in one part of your body, go to your bloodstream, and cause a change to happen in another part of your body. So it's how cells communicate. They don't talk, but they send out chemical signals that is their communication between each other. All right, next, D, cell-to-cell recognition. So we've talked about this a little bit. All right, let's look at the bottom cell here. So the bottom cell, it says here, Glyco Protein. This is what this is called. What is attached to it? You have this green thing. What is the green thing? It's not green on your paper, but it's green on mine. What is that? Sage?
Glucose chain?
Yeah, a small little glucose chain, a small little carbohydrate chain. It doesn't necessarily always have to be glucose, but it's oftentimes is. And so you have this small little carbohydrate chain of various sugars. And so that's when we say glycoprotein, glyco in front of anything is referring to a chain of sugar molecules, which is a type of carbohydrate. So what would we call a lipid with a chain of sugars on it? A glycolipid. So we do have lipids, that's the case here. So this happens to be a glycoprotein. I used the analogy when I talked about this at the in chapter 6 that the carbohydrate chains are kind of like ID tags, in that they help a cell to know if they belong to that particular individual. So for instance, if you have, I use the example of getting an infection. If you get a bacterial infection or something, how does your body know that the cells that are infecting you are not part of your body? You have to have a way to know that, hey, this doesn't belong to us. And how that works is in part due to the carbohydrate chains on there. So like carbohydrate chains on my cells may be different than carbohydrate chains on your cells, all right? And so it can vary within a species. It definitely varies from species to species. And sometimes it can even vary within the body. Like one cell type in your body might have a different carbohydrate chain than other organs or whatever. All right, so this is cell-to-cell recognition. So how do they recognize it? Is that other cells in your body have kind of receptor proteins that can kind of fit the shape of this glycoprotein and fit together. And so, and then if you don't have those receptors, then that's how they can kind of tell that it's foreign. All right, that's cell-to-cell recognition. Then we have intercellular joining. So the molecules, the proteins can actually bond together. So it's how one way that cells can stay adjacent to one another. And then on Friday we talked about how the extracellular matrix attaches cells, and it does that because it's attached to each cell and it attaches through these proteins. All right. So those are your functions of your proteins. So like I said, we're going to talk right now in about one slide about cell-to-cell recognition. And then we're going to talk a lot about this transport part of it, since we're talking about the cell membrane and getting things into and out of the cell. So the role of the membrane, carbohydrates, and cell-to-cell recognition. Cells recognize each other by binding to surface molecules, which are often carbohydrates, on the plasma membrane. These membrane carbohydrates are can be covalently bonded to lipids. We call those glycolipids, or more often to proteins forming glycoproteins. And carbohydrates on the external side of the plasma membrane vary among species, individuals, and even cell types in an individual. We'll talk later on in the course about how this plays a role in like organ donations and rejecting, and so on. All right, so to tie this in with the endomembrane system, with membranes have distinct inside and outside faces. The asymmetrical distribution of proteins, lipids, and associated carbohydrates in the plasma membrane is determined when the membrane is built by the ER and the Golgi apparatus. So let's look at the next picture here, which is the endomembrane system. And this is kind of like exactly almost what we did when we went over the homework that we went over on Friday with one of the questions. Remember that you had to trace the pathway? A lot of people had trouble with that. One of the questions was, where does a protein embedded in the cell membrane come from? And so this is what this picture is kind of showing you here. So here you have a the endomembrane system. So you have the ER, which buds off the transport vesicle, which goes to the Golgi, which buds off another transport vesicle, which goes to the cell membrane. And we talked on Friday about how when the transport vesicle binds to the cell membrane, the membrane that was part of the transport vesicle actually becomes part of the cell membrane. And so any proteins that are embedded in the membrane of the transport vesicle are going to become embedded in the cell membrane. So here we have like a Glyco protein, a protein that has a carbohydrate chain. Notice here it's in the transport vesicle, and when it fuses, this Glyco protein is going to become part of the cell membrane. So that ultimately, when we go backwards, this came from the transport vesicle, which got modified by the Golgi, which goes up to the transport vesicle, and was originally made in the ER. So therefore, in the ER, this protein got modified, was made, went through the transport vesicle to the ER, through some modifications. Now is in a membrane of the transport vesicle, which then becomes part of the cell membrane. So that's exactly what that question in our homework was about. All right, so that's where all these things come from, is the help of the endomembrane system that are part of the cell membrane. All right, so that's the structure of the cell membrane. So we're going to take another kind of look now at the job of the cell membrane. So now we know what it's made of, its main components. The cell has to exchange materials with its surroundings. That's a must. This is controlled by the plasma membrane. Plasma membranes are selectively permeable. They allow certain things to go in and certain things to go out of the cell. So let me ask you this. So we know the main construction. We have a phospholipid bilayer. We have proteins. We have cholesterol embedded in here. We have the carbohydrate chains hanging off some of the proteins and some of the lipids and so on. How is it that a membrane is selectively permeable? Like, the membrane itself, it's part of the cell. The membrane's not alive. It doesn't have a brain that says, oh, we're going to let certain things in and certain things out. So what in the world do you think plays a role in if something can get through the membrane?
Proteins would have like a little place for the certain thing, like the exact shape of what it wants to go in, and then maybe like the shape and nothing else.
Okay, so if you couldn't hear Nathan, he said the proteins might have a certain shape that only a certain molecule can bind, and therefore it only allows that certain molecule to go through because of the shape of the protein. Absolutely. Whether it's hydrophobic or hydrophilic. The main bulk of the phospholipid bilayer is hydrophobic. All right. So what do you think can get through the lipid bilayer most easily, hydrophobic or hydrophilic molecules? So, and realistically, when I draw the phospholipids, to give you an idea, like the phospholipid heads are much smaller proportion of the phospholipid bilayers. The fatty acid chains are pretty long compared to the heads. That's what I mean by the majority of the phospholipid bilayer is made out of the fatty acid chain, which is hydrophobic. So hydrophobic or hydrophilic, do you think could squeeze through here? Would hydrophilic molecules have any interest in coming through here?
No.
No. They'd rather stay out here with the other hydrophilic things, all right? And so therefore hydrophobic molecules can pass easily through the membrane, and hydrophilic molecules not as easily, all right? So absolutely. So whether or not the molecules are hydrophilic or hydrophobic, the shape, all right, also plays a big role, all right? So let's look at, actually, I'm going to have you look at the little sheet that you picked up here today, because this shows you visually what this is. So you have on the left a lipid bilayer. So this is supposed to be your phospholipids. And so this is what we have here. And so the interior, the core of the lipid bilayer is hydrophobic. So that's why if we look here, what can go through, we have hydrophobic molecules are able to go through, and it's showing you that by an arrow going right through the lipid bilayer, and that's because of that hydrophobic core through the membrane. And it gives you examples of what kinds of molecules are hydrophobic and can get through a membrane. So oxygen, nitrogen, benzene, and so oxygen very important for cells to get through. So it can go right through the lipid bilayer. Notice also the other types that can get right through the lipid bilayer are small uncharged polar molecules. So, polar molecules are polar molecules hydrophobic or hydrophilic?
Hydrophilic.
Hydrophilic, right? But the key here is that they are small. So we found that if the molecules are small enough, they can actually it will diffuse through this hydrophobic core. I'm going to have you cross off one molecule here. Let's cross off H2O. They used to think that H2O, because H2O is a small polar molecule, diffused through the lipid bilayer. We've since found out that that is not true, all right, which often happens in science. We figure out what we think was true was not true as we learn more things. All right, so those are the types that can go through. So notice the next two types. You have the arrow kind of ricocheting off. Can these guys get through the lipid bilayer?
No.
No. These guys cannot. So let's look at these guys. The only difference between the small uncharged polar molecule, large uncharged polar molecule, is that they are large. So if they're too large, they don't get through the lipid bilayer, and so they are rejected. And so glucose and sucrose are examples of those. Ions also, those have a big charge on them, and they cannot. They're very hydrophilic, and therefore cannot get through the lipid bilayer. But let me ask you this. Let's look at some of these examples. Glucose. Glucose can't get through the lipid bilayer. Do your cells need glucose?
Yes.
Absolutely. This is the molecule that is most used for energy from your cells. They have to get into the cells to be used. Certain ions we need, like calcium ions. Your muscle cells won't contract without calcium. We have to be able to get calcium in there. And so these guys have to get into the cell. How do they get into the cell?
Protein.
Yeah.
Receptor proteins or transport proteins.
Transport proteins. So these guys here, they can enter and leave the cell. Through a transport protein. It can't go through the lipid bilayer. So transport proteins are very important to get through these. And I'm going to add, while we have on here, I had you cross off water up here. Do you think water can get through the lipid bilayer, or through the membrane? Absolutely. We need to get water in our cells, or we die very soon. So we're going to add here, water they found is actually one that has its own transport protein. So water, as an example here, water is a unique situation. Water is a small, uncharged polar molecule. Small and polar. So that's why they used to think it belonged up here, because it's a small, uncharged polar molecule, so it made sense with that. But what they found is that there are special proteins called aquaporins that water actually diffuses through. All right, and so that's why I'm putting them down here. All right. Okay. So then let's look at our next one. So hydrophobic or nonpolar molecules such as hydrocarbons can dissolve in the lipid bilayer and pass through the membrane rapidly. But polar molecules such as sugars do not cross the membrane easily. When we say do not cross the membrane easily, we're talking about the lipid bilayer. They can cross the membrane if they go through a protein. So let's look at the proteins, the transport proteins. Transport proteins allow passage of hydrophilic substances across the membrane. Some transport proteins called channel proteins have a hydrophilic channel that certain molecules or ions can use as a tunnel. Channel proteins called aquaporins facilitate the passage of water. So, if I were to draw a transport protein, let's say this is, they literally have a tunnel, so I'm going to channel through, so I'm going to draw that here, through that substances can move through. So this is a transport protein. Let's say as an example, it is an aquaporin. So we just wrote about aquaporins. These guys specifically, example aquaporin. So these specifically let water through. So you have water, let's say, on the outside of the cell, and we want to get the water into the cell. Well, we just talked about how these proteins embedded in the membrane have a polar part that faces the water-based solution, and the interior is nonpolar. So, but the thing is with the channel protein is if this part in the middle was nonpolar, this water would never diffuse through. So when it says here the channel proteins, the way that they're constructed, there are groups that are polar make up the channel proteins here. So they make up, this part right here is polar. So the amino acids that have polar R groups are surrounding this channel. The rest of the molecule out here that's facing the hydrophobic tails is nonpolar, but that channel is surrounded by polar R groups. So therefore water can diffuse through and get into the cell through that polar channel. And that's what this is talking about here, have a hydrophilic channel that certain molecules or ions can use as a tunnel. Right? So I use the aquaporin as an example. It could be another channel that allows a certain ion through. So anything hydrophilic, that could be the case. So those are your channel proteins. Another type of transport protein changes shape a little bit. So this is not changing shape here. Other transport proteins called carrier proteins bind to molecules and change shape to shuttle them across the membrane. A transport protein is specific for the substance it moves. So let me give you another example here. I should say here, when I wrote transport protein, this is actually a channel protein because it has the channel here. I'm going to show you now how that differs from our carrier proteins. So let's look at another transport protein. So this is type 1, this is type 2. This is called a carrier protein. I'm going to draw it here to give you an idea of what this means. So a carrier protein, let's say here, has a certain shape, and I'm going to draw here the shape like this. Whoops. All right, the shape is important. So let's say inside the cell you have two molecules. You have molecule type, we'll call it molecule A, and you have another type of molecule that's shaped like this, molecule B. Each molecule has a unique shape. Do you see that the construction and the shape of this carrier protein fits nicely with molecule A? So molecule A can come over here and bind to these sites here. Molecule B cannot. So therefore the carrier protein is very specific to what molecule it's going to quote unquote carry across the membrane. So once these molecules that have a specific shape that's complementary to the shape of the carrier protein binds to it, the carrier protein actually changes shape. So what happens is when it changes shape, it's going to open along the other side, so it's going to kind of be upside down like this. And when it changes shape, it can actually change the shape of where the molecule actually was bound. That's important. So now I tried to make pointy shapes. So this was bound to this. Now that the protein has changed shape, molecule A is going to get kicked out of that bonding site and will leave and go outside the cell. And so therefore, this carrier protein has carried this molecule from one side of the membrane to the next. And this is why, because the shape is important, this is why it's very specific for the substance it moves, all right, because of the shape. So those are your two ways to get things across the membrane through a protein. What's your third way that something can move across the membrane? One of these two ways, or directly through the lipid bilayer. All right, so it can go directly through the lipid bilayer, or one of these ways through a protein. All right, so that's what we're going to look at is why they move across. So we looked at the how here, where they physically move across the membrane and what kind of molecules move where. Passive transport. The diffusion of a substance across the membrane with no energy investment. Diffusion is the tendency for molecules to spread out evenly into the available space. Although each molecule moves randomly, diffusion of a population of molecules may exhibit a net movement in one direction. At dynamic equilibrium, as many molecules across the membrane one way as cross in the other direction. So in looking at this, that first bullet when it says diffusion is the tendency for molecules to spread out evenly into the available space. I'm going to have you add behind that when molecules diffuse, they move from an area of what concentration to what concentration? You guys remember?
High to low.
High to low. So they move from an area of high concentration to an area of low concentration. And so jot that down. That's always the case with diffusion. And it doesn't require any energy. So that's what passive transport means, is that substances are being transported, but you don't have to put any energy into it. It does it naturally. So let me give you an example that's not with the cell. All right, well, if I were to just stand up here with a bottle of perfume, and I sprayed the perfume molecules into the air, so I squirted it once, I would obviously smell it first. Who would smell it next? The people right nearest me, right? Would the people in the corners, in the back, eventually be able to smell it? Yes. So my question to you is why that is. So what scientifically is going on with these molecules that would be the case, that they would move? So I'm going to give you a second, I'm going to have you talk in your group at your table about why that is. Why would it move like that?
They don't want to be next to each other. They're diffusing from higher concentration to lower. They're more concentrated when it comes out at first, and then they spread out to get away from this.
Let me point something out. When I have students do this a lot, this is sometimes what I get as an answer. Because they want to move to an area of low concentration. Do these molecules, do they have a thought? Do they have a thought? No, they don't want anything, all right? So that should not be part of, maybe that's what they're gonna do, that's the ultimate end result, but the reasoning behind it. Also, it's like they want to diffuse from an area of high concentration, so no wants in this, all right? So scientific principles, what's going on that these molecules start up here and end up in the back of the room, all right? So, all right, continue conversation. Okay, so no wants in the answer.
Air is a fluid.
What does that mean in regards to
confident about your group's answer, or who's willing to share. Maybe you don't have to feel confident about it. Who wants to share the reasoning? Shane?
Okay, so air molecules go between all the protein molecules, creating more space between all the protein molecules.
Okay. When the air molecules go between them, what do you? They're
Mixing. That's why.
and they're mixing why?
Because of like, I don't know, the charges in it, or because they know we're not.
They do mix. They do mix. I'm just trying to get at the why do they mix. So it's not like the air molecules are like, ooh, perfume molecules, get away from each other. Right? They don't do that. So what's going on? Why do they mix? Joe, you have an idea? No? Devin, you have an idea?
Not really.
Maddie?
Don't the perfume molecules not attract to each other because they're the same molecule, and the air moves, or the air molecules move with such high intensity because they're air molecules that they're pushing them away, and the perfume molecules don't really bond well?
Well, just because they're the same molecule doesn't necessarily mean that they don't bond. Like water is an example. That water molecules bond with hydrogen bond with each other. So it actually has nothing to do with them bonding with each other. Or not bonding with each other.
Is it like the density of the molecule?
Anybody have any other? So when they mix, what do they have to do? When you spray it in here, you use the word mix. For the air molecules to mix with perfume molecules, what do they have to physically do? Move, right? How do molecules move? Every molecule has kinetic energy. They're bouncing around. Like the air in here is not static. Like the oxygen molecule that's right here that I can't see doesn't stay right there. All right, it moves around. And so these molecules have this kinetic energy. Now you spray these new molecules into the air. These perfume molecules now are in the air. They are mixing, but why are they mixing?
Is it because it's either the perfume has more kinetic energy because you're like forcing it out, or the air molecules do because they're moving faster, and therefore like pushing.
So it's a little bit of both. They both have their own kinetic energy. So these new perfume molecules have this kinetic energy. All the air molecules, oxygen, nitrogen, there's water molecules in the air. All of these guys have kinetic energy. And so what happens is as they move, they hit each other and bounce and ricochet off of each other and so on. And pretty soon, given time, some of these perfume molecules will make their way over here. They're not going that direction. They are going a little bit that direction, and then the wall stops them, right? And so eventually, so the net movement is over this way, and eventually the people in the back would be able to smell it as they slowly move and ricochet and bounce off of one another, so that eventually, no matter where I went in the room, it would pretty much smell equally, all right? And that's because you get to a point where those molecules have bounced off each other, that they're pretty much what we call an equilibrium or an equal concentration no matter where you go in the room. And that's what we call equilibrium. So as many molecules cross in one way as the other way. So that means that once it reaches equilibrium in this airspace, that means if a molecule ricochets and moves this way, because there's an equal concentration of them, most likely there's another one that's moved in this way. And so no matter where you go, it's an equal concentration. And so that is diffusion. We had an area of high concentration here to an area of low concentration, but the reason has to do with the random movement of the particles. All right, and why do they go from high to low? Because you just have more molecules here diffusing and ricocheting off of them, all right, and they only have one place to go. All right, so this picture here shows you diffusion in a liquid. So the picture on the left is showing you that you have dye molecules dissolved in water. So the blue in here is water. They're not showing you the individual water molecules. And the left side is separated from the right side by a selectively permeable membrane. This is a little bit different. This is a fake membrane. It's not exactly like the cell membrane. This membrane is selectively permeable based upon size. It has pores in it. And basically, if the molecule is small enough to get through the pore, it can go to the other side. So what happens is, when we look at this, we can clearly see that the higher concentration of dye is on the left than the right, because there's no dye on the right. So you should be able to tell me that it's going to go from left to right. Why is it doing that? Because of the random movement of the dye particles. So the dye molecules randomly move. If they happen to hit an open pore, they can move to the left-hand side. Are they always going to hit an open pore? No. So sometimes that's what this one's showing you here. They hit the membrane and it ricochets back off and it stays on that side. But so what happens is you're going to eventually end up with some molecules on the left. Does it mean that the molecules on the left, or sorry, the right? When you look at the molecules on the right, they're still moving, but you still have your higher concentration of dye on the left. So what happens is these guys are still going to be moving, randomly hitting the open pores and still diffusing in this direction. These guys, can one of them randomly hit and go in this direction? Absolutely. And so that's what this right here is showing you here. But because there's so few dye molecules here, the chances that the statistics that they're going to hit a pore and go to the left is much lower than the chances that these guys are going to hit a pore and go right. That's what we call net diffusion. Net diffusion is going from left to right. It doesn't mean that there's no diffusion of the dye going to the left-hand side. There is some, but because there's an overwhelmingly greater diffusion going from left to right, we call that the net diffusion. And so eventually equilibrium is reached. Do the molecules stop moving at equilibrium? Absolutely not. They're still moving, so you still get diffusion back and forth, but if it's in equilibrium, your concentrations are roughly equal, so therefore the chance of a molecule hitting and going to this side is equal to that hitting and going to this side, so they go back and forth at equal rates, all right? And so that is what equilibrium is. And so that's across the membrane with the molecules of dye. So we say, when we're talking about substances diffusing, that substances diffuse down their concentration gradient, which is the difference in concentration of a substance from one area to another. No work must be done to move substances down the concentration gradient. The diffusion of a substance across the biological membrane is called passive transport because it requires no energy from the cell to make it happen. So my perfume example, the perfume molecules diffuse down their concentration gradient from an area of high to an area of low concentration, down the concentration gradient. And I didn't have to make that happen. You don't have to put any energy into it. When you spray perfume in a room, it happens all the time when people put lotion on in rooms and so on in classrooms. Eventually you smell it and the whole class smells of it. The person doesn't have to do anything. They're not like blowing the perfume molecules around. It happens with the natural kinetic energy. And so, so that's what it means by no additional work. So back over here, we went down the concentration gradient from high to low concentration. So whenever substance is doing that, from going from high to low concentration, it doesn't require any additional input of energy. All right, and this picture just shows you that two solutes in the same scenario as the previous picture, but showing the purple solutes. The purple solutes are in high concentration on the right-hand side, and they're going to be moving and doing their own thing. And since there's a higher concentration of them here, they're going to go from right to left. So therefore, the net diffusion here is from right to left, because even though as they diffuse over to the left-hand side, there's so few of them that the likelihood that they hit is much smaller than the ones on this side until eventually equilibrium is reached. And both of them are separately in equilibrium. All right, so that's that. So that's the diffusion of particles. The diffusion of water has a special name that hopefully you remember, is osmosis. The diffusion of water across a selectively permeable membrane. The direction of osmosis is determined only by a difference in total solute concentration. Water diffuses across the membrane from the region of lower solute concentration to the region of higher solute concentration. Solute, what is solute?
Solute is what's dissolved into the water or any.
That's right.
Yeah.
All right. So what is dissolved in there? So when we talk about osmosis as the diffusion of water, notice here we're looking at the total solute concentrations or what's dissolved in the water to figure out where water is going to move. So since osmosis is the diffusion of water, diffusion we just said is going from what to what concentration? High to low. So water is going to go to from high to low, but we use the solute concentration of the solution to determine where the higher water concentration is. And so this picture here, this picture is a piece of glassware called a U-tube. And so on the left-hand side is the original U-tube. On the left-hand side is what I want to focus on. When you're looking at this, it's separated, the two sides are separated by a selectively permeable membrane. The volumes of water are equal on either side. That's what the levels are, equal levels are supposed to be showing you. Selectively permeable, it tells you sugar molecules cannot pass through the pores, but water molecules can. So that means this is selectively permeable based upon size. So the sugar molecules are too big, can't move, but water molecules can. Each one of these little balls are supposed to represent a sugar molecule. So if you have equal volumes of water, you can see that there's more sugar dissolved in the water on the right than there is on the left. So there's a higher concentration of sugar on the right than there is on the left. In this picture then, what I want you to do is label the side that has the higher water concentration. On the picture on the left, which side has the higher water concentration? When I mean by side, I'm talking about this first U-tube. On the left side of this U-tube or on the right side of this U-tube, which one has the higher concentration of water? And this is where we'll stop. We'll pick this up tomorrow. You guys remember diffusion and osmosis at all? A little bit.